Hello, everyone, and welcome to the second half of our look on Chapter 11. During the first half last week, we went through the evidence of evolution. In the second half, we're going to look at the processes of evolution, meaning how does evolution actually take place? We've seen the evidence that shows us that it's happening, but how does it actually work? And a good place to start is by reminding you of the definition of evolution. So, in our last lecture, we defined evolution as the fact that populations of organisms change over time, becoming genetically different from their ancestors. So, basically, over long periods of time, living organisms change. That's evolution. But why and how does this happen? That's what we're going to tackle in this lecture. It's often said that variation is the raw material of evolution. Evolution is change over time. And for that to happen, there has to be variation. There have to be differences that exist in the genes and traits of a population. All types of organisms, from plants to animals to microscopic bacteria, they all exhibit genetic variation. And this paves the way for populations of those organisms to evolve. But where does that variation come from? What causes it? Well, one way that we get variation is through sexual reproduction. Not all species engage in sexual reproduction. Some, like bacteria, basically just clone themselves to reproduce. But among species that do reproduce in a way that involves two separate partners, they end up creating unique genetic combinations in their offspring. But regardless of whether you are a sexually reproducing species or not, all species have another source of variation that causes changes to crop up, and that is mutations. Mutations, as we talked about back in Chapter 9, are changes in DNA. And of course, these lead to new variations on characteristics. Mutations can be caused by many different forces. They can come about when a living organism is exposed to some sort of an agent that damages its DNA, like chemical toxins or radiation, but they can also be caused simply by random mistakes that take place during cell reproduction when the cell is duplicating its DNA and it accidentally adds the wrong letter, or it adds too many letters, or it misses a letter. So mutations are inevitable even in the absence of exposure to things that would damage DNA. They happen in all living things. The rate at which mutations happen varies from species to species, though. So, for example, in the bacteria known as E. coli, there is about one mutation per 1,000 generations, which seems like a very long time. But because E. coli are bacteria and they reproduce so quickly, 1,000 generations is only about 14 days in the time frame of E. coli. Stale cress, which is a type of mustard green that we've mentioned once before, it has about one mutation per generation that sticks in the lineage. And then humans have somewhere in the range of 10 to 100 mutations per generation. So, what this means is that if you took all of the DNA that you got from your mom and you compared it to her DNA, and you took all of the DNA that you got from your dad and compared it to his DNA, 10 to 100 of the letters in there wouldn't match. They would be new or different compared to what's reflected in your parents. So, mutations are happening all the time, and although most of them are minor, some of them do lead to new variations on traits. So let's look at one familiar example, which is achondroplastic dwarfism in dogs. So achondroplastic dwarfism is a condition in dogs caused by a mutation in the fibroblast growth receptor gene, growth factor receptor gene. And fibroblasts are a type of cell in the body that contributes to the formation of connective tissues like tendons and ligaments. And the mutation in this gene causes abnormal bone growth and disproportionately small limbs. So, dogs that have this mutation have legs that look way too short and stubby for their bodies. So, some breeds that have this mutation are dachshunds, corgis, and basset hounds, which you can see on this slide. And the molecular biology of this condition has been very well studied. And the extraordinary thing is that it's the same mutation that causes this dwarfism in all three of these dogs, plus 16 other short legged breeds that we know of. It's a recessive mutation, but these breeds have been selectively bred to only include that recessive allele in their lineage. So if they're purebred, then they always have short legs. And on top of that, this same dwarfism mutation has been found in the DNA of wild wolves in Europe and the Middle East. However, because it's recessive, it's pretty much hidden in the wolves. The wolves are only carriers of the allele. So, what this means is that at some point, one of the ancestors of domestic dogs experienced a random mutation that created the allele for dwarfism. And that allele kept getting passed down. And when humans started domesticating dogs and breeding them together, they discovered that you could create these stubby little breeds that have two copies of that mutation. And just like with Shrek the sheep, this mutation wouldn't do much good for these dogs in the wild, but for human purposes, it can be beneficial. So, for example, this drawing is of a turnspit dog, which is a breed that was relied upon in the 1500s through at least the early 1800s to run on a small wheel that would cause a rotisserie spit to turn over a fire and cook meat. And on the right-hand side of the slide, you can see an illustration of a turnspit dog running a wheel that is anchored to the ceiling. So, under these circumstances, having short legs and a long body was a benefit, and humans latched on to that dwarfism mutation and bred dogs for this purpose. Nowadays, of course, people don't breed dwarf dogs as working dogs to turn a rotisserie spit, but rather as pets because we think they're cute when they're stubby. And this has led them to become more common than ever before. So we could ask ourselves the question, what percentage of dogs across the world were affected by achondroplastic dwarfism 10,000 years ago? Probably very few. But today, after thousands of years of selective breeding and developing this cultural obsession with stubby dogs, the representation of dwarf dogs in the world population of dogs has skyrocketed. Corgis and Dachshunds are some of the most popular breeds in the US. And this is evolution. Evolution is genetic change on a population level. One individual organism having a mutation is not evolution. But when the gene pool is significantly transformed by a change in how common a particular variation is, that is evolution. So this mechanism of evolution that we've been talking about, that's called artificial selection, also known as selective breeding. It's the process of humans choosing to breed individual organisms that have desirable traits in order to ensure that those genes get propagated and continue onward in the gene pool. And we talked about this with Shrek the sheep. We've talked about it now with dwarfism in certain breeds of dogs. But really, it applies so much more broadly to all kinds of traits seen in all kinds of organisms. The wide variety of traits that we see in modern breeds of dogs is all attributable to artificial selection by humans. So, identifying a mutation that we liked and then making sure it gets passed on. And this has also been done with plants. Did you know that 6 of the vegetables that we eat are actually all the same species of plant, which has been selectively bred to enhance different traits? So, Brussels sprouts, cabbage, kale, broccoli, cauliflower, and kohlrabi are all the same species, Brassica oleracea, but they are distinct lineages of this species that have been selectively bred in different ways. Brussels sprouts are the result of breeding Brassica to enhance the size of its lateral leaf buds. Broccoli is the result of breeding it to enhance the flower buds and stems. Kale is the result of breeding it for the leaves. So you can see how over many generations, a species can really be transformed when you're selecting certain mutations to carry on. But of course, this is how it works when humans are pulling the strings, which does not tell us about how the process of evolution works in nature, in the wild, when humans don't have their hand in it. So let's talk about that. What causes evolution to happen in nature? There are 4 major mechanisms by which evolution can take place in the wild. Those are natural selection, genetic drift, and gene flow. So in this lecture, we're going to take a look at how each of these works one by one, starting with natural selection. Natural selection is the more abundant reproduction of individuals whose traits allow them to survive better in their environment. It's based on three principles. First, variation. As we know, within a population, there is variation. Not all organisms have the same traits, and inevitably, organisms with certain traits will be better suited to survive in their environment. Second, competition. In nature, organisms must compete for limited resources, and only some survive. So there's a competitive pressure. Not all organisms are going to make it. And then third, putting these together, organisms with fitter traits will have more opportunities to reproduce and for their offspring to inherit those fitter traits, which means they become more represented in the gene pools of the future. If this sounds familiar to you, it's because it should sound familiar. It sounds a lot like artificial selection, sounds a lot like selective breeding, and it is just like that, except nature is the breeder instead of humans. So instead of humans dictating which organisms reproduce based on which ones have traits that we think are desirable, in natural selection, nature dictates which organisms reproduce by virtue of which ones have traits that allow them to survive. Organisms with greater fitness survive better and, by extension, breed better, which increases the representation of their genes in the next generation. And just like how we can see that humans have selected for a wide variety of traits in modern dog breeds, we can see how nature has selected for a wide variety of traits in wild dog breeds. For example, take the coyote. This is one type of wild canine. You can see how it has a coat that allows it to blend in and camouflage with its environment, as does the dingo, which is another type of wild canine that lives in a different environment. You find the dingo in the Australian outback. It's also the case for the Arctic wolf, which, as the name suggests, lives in the Arctic, uses its white coat for camouflage, and also for the African wild dog that lives in the grasslands and the savannas of southern and eastern parts of Africa. So all of these are wild canines that came from a common ancestor, but they all look very different because in these different environments, being well suited for survival and being physically fit, it doesn't look the same in each of these environments. So let's talk a little bit more about this idea of fitness. Because when we talk about fitness in the context of natural selection and evolution, how fitness is really measured is by reproductive success, or in other words, the number of offspring. Fitness does not necessarily mean that an organism is the largest or the strongest or the most athletic. In evolutionary biology, the most fit organism is the one that has the most success at making offspring. And this is dependent on their survival ability, because better survival means more opportunities to mate, which means more offspring and a bigger contribution to the next generation's gene pool. With that in mind, I have your first checkpoint for you. Which is the fitter individual according to natural selection and why? Is it Bob, a Harvard-educated neuroscientist and marathon runner who has 0 children, or Billy, who is unemployed, suffers from type 2 diabetes, and has 12 children? So we have an understanding now of how natural selection works. And so it should make sense to us that natural selection can lead to the evolution of adaptations. An adaptation is an inheritable trait that helps an organism's survival and reproduction. So take, for example, the desert hare. The desert hare has evolved long, flat ears with big blood vessels, and you can even see the blood vessels in this photo. And these ears allow them to efficiently cool their bodies. Blood moves through the vessels in the ears and it releases body heat into the air around them, which is very beneficial for a species that lives in hot desert environments. So these ears provide a survival advantage. But on the other hand, now let's take a look at a cousin of the desert hare, the Arctic hare. The Arctic hare's ears look nothing like the ones that you see in the desert hare. The Arctic hare doesn't need to cool its body efficiently, it needs to keep its body warm. So instead, it has short, stubby ears that are close to its body and insulated with fur. If an Arctic hare were to be born with ears that looked like a desert hare's ears, it would probably die and it would probably never get the chance to mate and have offspring, and vice versa for the desert hare if it was born with a mutation that gave it those short, stubby ears. So this brings us to our next point, which is that there are no objectively fit adaptations. Traits are only adaptive in a specific environment. There's no such thing as a good trait or a bad trait, full stop. Whether a trait is beneficial and adaptive depends upon what the conditions are like where the organism lives. Let's look at another example, the katydid. Katydids are insects that are found broadly across all continents except Antarctica, and some of them have evolved an appearance that is strikingly similar to a leaf, as you can see in this graphic here. This, of course, provides them with camouflage from predators and enhances their survival, but only in certain environments. So if you take a katydid that looks like the one in this video clip and you drop it into the Sonoran Desert, is its appearance going to be considered an adaptation any longer? No, it'll stick out like a sore thumb and it'll get snapped up by the first insect-eating animal that sees it. Instead, in the desert, katydids look like this, what you see on the bottom. So that's much better, right? That's much more adaptive. This is the product of natural selection in action. When nature is the breeder, it breeds organisms with traits that help their survival and reproduction, at which point they're considered adaptations. So now a couple more checkpoints for you. I'd like you to use your imaginations and describe an environment in which having a long, pointy beak would be considered an adaptation for a bird. And then for checkpoint 3, describe an environment in which a long, pointy beak would not be an adaptation. So, how do we know that natural selection happens? Where do we see observational evidence of natural selection happening in the wild? One area, which we've mentioned several times before, is in antibiotic-resistant bacteria. We talked about this in our last lecture. The proportion of bacteria that are resistant to the medications that we use to kill them is growing over time. The reason why this is the case is because occasionally, due to randomness, bacterial cells can develop mutations that make them resistant to antibiotics. So when a person takes a course of antibiotics to treat an infection, if one of these mutant bacteria is in their body, it will survive, while its fellow bacteria that lack that beneficial mutation will die off. And this means that the mutant bacteria will get the opportunity to reproduce and the mutation gets passed down. And because now all of its friends are dead, its offspring makes up a big proportion of the next generation. And so their population increases. And this process is inevitable. It means that over time, bacteria are going to develop resistance to our antibiotics. There have been multiple documented cases where a person has died from an infection that is resistant to every antibiotic that exists in our medical system. So this is why the fight against bacteria has been referred to as an endless arms race, where pharmaceutical scientists are constantly trying to develop new antibiotics as bacteria undergo natural selection and become resistant to our old ones. And it's why it's so important to only take antibiotics when you really need them, because taking them for minor infections only creates the further opportunity for bacteria to evolve, and it gives them more advantages in that arms race. Another very similar situation where we have evidence of natural selection taking place is in agriculture, specifically with weeds that are becoming resistant to weed killer. The most popular weed killer used in commercial agriculture in the US is called Roundup. You may have seen it at plant nurseries or hardware stores. The active ingredient in Roundup is a chemical called glyphosate. And on the bottom of this figure, in that little black rectangle, you can see it labeled glyphosate resistant species. So this key at the bottom tells you how many species of weeds that are resistant to glyphosate have popped up over the years in various states. And you can see how in the early 2000s, only a few states had 1 or 2 species of glyphosate resistant weeds. But within 10 years, the problem has become widespread throughout ag country. This is a real problem. And the source of the problem is exactly the same as the antibiotic-resistant bacteria. Once in a while, a weed emerges that has a random mutation that makes it so that glyphosate cannot kill it. When farmers spray their field with Roundup, other weeds die, but the mutant weed survives, which means it gets to reproduce, and then it contributes to the next generation, so the mutation spreads. So those are two pieces of evidence that show natural selection in action. And these are some pretty extreme examples of how particular mutations are increasing rapidly in prevalence. And they may lead you to wonder, why doesn't natural selection cause the fittest form to eliminate all others? If you take natural selection to its logical end, won't the most fit trait eventually just come to dominate the population and you'll be left with no variation at all? And sometimes that does happen. However, in most cases, it does not, and there's several reasons why. First, the conditions in the environment are always changing, and this means that as new challenges are thrown at a population of organisms by the environment, they're going to have to develop adaptations in a different direction before one variation has the opportunity to take over. Also, many mutations bring about traits that are simply neutral. They aren't beneficial to an organism, but they also aren't detrimental. And in that case, there's nothing working to eliminate those traits, so they can just sort of putter along in the gene pool and contribute to new variations. And then finally, sometimes, even if a particular trait is detrimental, being a heterozygous carrier for that trait may have an advantage. When this happens, it's known as heterosis. One very well-studied example of heterosis actually takes place in humans in relation to sickle cell anemia. So we just learned about sickle cell back in chapter 9. But to remind ourselves, I have a checkpoint for you. What is sickle cell anemia and what causes it? So now that we've refreshed our memory on what sickle cell is, I'll also remind you that having sickle cell anemia requires 2 copies of the sickle cell hemoglobin allele. A person needs to have the sickle cell allele on both of their chromosomes. They have to be homozygous. They have to have gotten it from their mom and their dad to be affected by the disease. We also learned that individuals who have just one copy of the sickle cell hemoglobin allele, meaning they're heterozygous, they got it, for example, from their mom but not from their dad, they are considered to have a condition known as sickle cell trait. And these individuals are mostly symptom-free, but elite athletes with sickle cell trait have a higher risk of things going wrong when they're performing at a high aerobic capacity. But despite being heterozygous being a problem for elite athletes, people who are heterozygous for the sickle cell allele are actually also protected from a deadly disease called malaria. Malaria is caused by a parasite that is transmitted from mosquito bites, and the most severe species of this parasite is called P. falciparum, and it's a parasite that infects red blood cells. And in this image, the darker purple rings that you see inside of the red blood cells, those are actually the parasite. Hundreds of thousands of people are killed by malaria every year around the world. Most of them are children. About 500,000 children are killed by malaria each year. But being heterozygous for sickle cell has a protective effect. The reason why is because when the malaria parasite enters the red blood cells of a person who is heterozygous for sickle cell, although the cells are initially normal in shape, the entry of the parasite triggers them to immediately become sickled. And when this happens, it sounds a very obvious alarm to the white blood cells in your body, which go on the attack and destroy the sickled red blood cells that have been infected with P. falciparum. So for people who do not have the sickle cell allele at all, this very obvious sickling reaction to the infection doesn't happen. And so the white blood cells don't immediately recognize the infection and fight back against it in such an efficient way. So this is heterosis. Being homozygous means you have sickle cell anemia, and that's definitely a disadvantage. But being heterozygous means you have a survival advantage against malaria. And this is why the sickle cell allele has been maintained in the gene pool in sub-Saharan Africa. So on the left-hand side of this figure, you can see a map of the African continent showing the rates of sickle cell allele. And on the right, you can see a map showing the range of where P. falciparum, the parasite, can be found. And the overlap between these two ranges is strikingly similar. They match each other very well because the areas where you find this severe form of malaria is the area where sickle cell allele gives you an advantage. So sickle cell hasn't been eliminated from the gene pool for that reason. So that covers our discussion of natural selection. But natural selection is not the only mechanism by which organisms evolve in the wild. We also need to talk about these three other mechanisms, although these other three are going to go by much more quickly. The next one is sexual selection. Sexual selection is the phenomenon where organisms that are better at securing mates have more opportunities to reproduce and pass on their genes. And often, this can actually be at the expense of their potential to survive. Among many species, there is a trade-off between attracting mates and being better at survival. Like, for example, in peacocks. Male peacocks have very bright and obvious tail feathers that they grow to impress their female counterparts. And of course, those tail feathers are the opposite of camouflage. They make them more obvious to predators, but they make them more attractive to females. Another example, among elephant seals, having an extremely large body size and strength is considered a benefit because only one male has the privilege of mating with the entire harem of female seals. And he has to fight off the other males for control of the harem. Oftentimes, the other males aren't killed. They're still part of the colony, but they simply just don't get to mate with any of the females, so their genes don't get passed on. So the message here is that individuals with more attractive traits become more represented in the next generation. Next, after sexual selection, is genetic drift. Genetic drift is the alteration of a population's gene pool by the effect of chance. Because sometimes random events happen that have nothing to do with the survival ability or the attractiveness of an organism. So let's say there's a big landslide that comes down and kills half of a population of wildflowers, but not the other half. This has nothing to do with how fit the individual flowers were. They just happened to be in the wrong place at the wrong time. Or let's say a wildfire burns through a forest and kills most of the deer population. Again, just the wrong place at the wrong time. So as you can imagine, small populations are much more susceptible to alterations of the gene pool by chance events. Because when there's fewer individuals and something goes wrong, there are fewer left to pick up the pieces and start fresh. If a landslide kills 100 wildflowers out of a population of 200, then that's, boom, instantly half the population is gone. But if it kills 100 wildflowers out of a population of 20,000, that's really just a drop in the bucket. So the small population is more significantly affected. So with that in mind, there are two common types of genetic drift. The first one is called the bottleneck effect, and the second is called the founder effect. The bottleneck effect is very much along the lines of the example that I gave involving natural disasters. It's a dramatic shrinking of the gene pool due to a random event that wipes out a large portion of the individuals in a population. The reason why it's called the bottleneck effect is because of the shape of a bottle. The body of the bottle is big, but the neck is narrow. And so when a population goes through a bottleneck, the symbolism is that only a few individuals make it through to the other side and survive. One real-life example of the bottleneck effect is seen in the case of elephant seals. Before the 1800s, there were hundreds of thousands of northern elephant seals living in the northern hemisphere. And then, due to them being extensively hunted by humans for their blubber, which was used to make lamp oil, their population was brought down to less than 100 individuals total worldwide. And actually, they were believed to be entirely extinct for years until a very small group of them was found on an island off the coast of Mexico in the late 1800s. Eventually, it became illegal to hunt them, and because of those laws, the population has rebounded back to almost 200,000 elephant seals today. But every single one of those 200,000 are descended from the fewer than 100 that managed to avoid being killed by humans during the hunting spree. The other type of genetic drift is the founder effect. The founder effect is a reduction in genetic variation within a new population founded by a small number of individuals who are separated from a larger population. So in this illustration, what you see is a few organisms that set off on their own. This illustration shows butterflies, so maybe they were blown off course in a windstorm, and now they have to start a new population, so they're no longer intermixing with the old population. So the gene pool is a lot smaller in this new population compared to in the old one. One really interesting example of the founder effect that can be seen in humans is among the Old Amish order in Pennsylvania. And this is an example we also mentioned at the beginning of our last lecture. The Old Amish order community was founded by a small number of individuals who immigrated from Europe in the early 1700s. And one of these individuals was named Samuel King. He immigrated with his wife in 1744. And King was unknowingly a carrier of the disease Ellis Van Creveld syndrome, which causes dwarfism and extra fingers and toes. And although Ellis Van Creveld syndrome is very rare in the world at large, worldwide it only affects 1 in 200,000 babies, the incidence of the condition in the Old Amish order community is 1 in 200. So it's 1,000 times more common. And this is because King passed down his mutation to some of his children, who passed it down to their children, and as more generations passed, cousins of varying degrees intermarried within this very small community. And today, the vast majority of Old Amish order members can trace their heritage back to those early settlers, and many of them are distantly related in some way to Samuel King. So because the community was founded by and descended from only a few individuals, the genetic abnormalities of this one founder have this big outsized effect on the population's gene pool. So that's genetic drift. The last mechanism of evolution we have to talk about is gene flow. Gene flow describes the flow of alleles in and out of a population due to the migration of individuals, their gametes, or their genes. Basically, it's the introduction of new material to the gene pool from individuals outside of the gene pool. One example of gene flow in humans is the contribution of some genetic material from Neanderthals to the modern human gene pool. So Neanderthals are the closest cousins of the human species, and they're now extinct. They shared a common ancestor with humans somewhere between 700,000 and 300,000 years ago. And although they had some physical features that were different from humans, as you can see in this illustration, they were so similar to humans that they are actually technically considered the same species. They're just different subspecies. So while modern humans are classified as the species Homo sapiens and the subspecies sapiens, we are Homo sapiens sapiens. Neanderthals are also Homo sapiens, but they're the subspecies Neanderthalensis, so Homo sapiens neanderthalensis. And Neanderthals were highly intelligent. They used fire, they built shelters, they made and wore clothing, they made art, they buried their dead, and they marked the graves of their dead. All of these things are what set humans apart from other apes in their evolution. And there have been hundreds of individual remains of Neanderthals uncovered from various sites throughout Europe and parts of the Middle East. So in this map, you can see the geographic range where Neanderthal remains have been uncovered, highlighted in blue. And these very same geographic regions were occupied by humans at the same time. Because Neanderthals didn't exist that long ago, the youngest ones are less than 50,000 years old, Because they're so recent, the DNA in many of these Neanderthal remains has been well preserved and recovered for analysis. And when you take the DNA from those remains and you look at the sequence and you compare it to the DNA of modern humans, what you see is that Neanderthals and humans must have interbred with each other after humans migrated out of Africa 100,000 years ago, which means that some of the modern human gene pool is Neanderthal in origin. Specifically, about 2% of the DNA of non-sub-Saharan Africans is Neanderthal in origin. If you look at the DNA of people who live and have always lived in sub-Saharan Africa, they lack any Neanderthal DNA because they are descendants of human communities that remained on the African continent and never migrated out to Europe where the interbreeding took place. Analysis shows that having Neanderthal DNA is associated with certain variations in skin color, certain variations in height, hair color, a higher tendency towards daytime napping, a higher risk of narcolepsy, and a higher risk of mood disorders, among other traits. So this is an example of how certain genes, certain traits flowed into the human population through breeding with Neanderthals. Another example of evolution by gene flow is seen in bacteria. Research has shown that some of the mutations that provide antibiotic resistance have been transferred across species from MRSA to other bacteria. So you may have heard of MRSA before. It stands for methicillin-resistant Staphylococcus aureus, and it's the cause of what's known as a staph infection, among other types of infections. And MRSA has mutations that make it resistant to members of the penicillin family of antibiotics. And MRSA has shown the ability to actually physically transfer those pieces of DNA to other species of bacteria, which then makes them resistant as well. So this is another example of how gene flow facilitates evolution. So now let's tackle a few checkpoints to apply this information in context. The male swamp sparrow performs these trilled songs to attract mates. Studies have shown that female swamp sparrows prefer males that perform more challenging songs that require rapid and precise coordination of their vocal tract movements. The evolution of this complexity has been a result of which of the mechanisms of evolution that we just talked about? Next, which of these two species would be more susceptible to the effects of genetic drift? Would it be San Francisco pink groundsel, which is a plant that only grows in the 2 square mile alpine tundra region of the San Francisco peaks above Flagstaff, Arizona, and nowhere else in the world? Or would it be dandelions, which grow widely across all continents of the Earth except for Antarctica? And lastly, Sika deer, which you see on the left-hand image, were introduced to Europe from Asia in the 19th century. They started crossbreeding with the native European red deer, which is in the middle picture, and this crossbreeding is producing significant populations of hybrid deer, which you can see in the right-hand picture. And these hybrid deer have characteristics of both species. So this is an example of evolution by what mechanism? So we now understand the mechanisms that drive evolution to take place in the wild. And we've seen some examples of how these mechanisms of evolution lead to changes of characteristics within species. However, evolution doesn't just cause change within species, it also leads to the emergence of new species. And we're going to talk about how. But first, we need to define what a species actually is. What makes a species? This question is actually more complex than it might first appear, because there are 3 separate schools of thought in biology on what makes a species a species. The first school of thought is that species are defined based on the physical characteristics of the organism. Second one is that the role that the organism plays in the environment is what defines a species. And the last one
is called reproductive isolation. Only organisms that can breed together are members of the same species. Now, all of these are used by biologists under different circumstances, but each definition comes with its own strengths and weaknesses. For example, if you choose to define a species based on physical appearance, or in other words, morphological similarity, morphology is the science term for physical characteristics, then what you're basically saying is that organisms that physically resemble each other belong to the same species. Now, the advantage of this definition is that it was the first and earliest species concept that has now been used for hundreds of years. But it has a major disadvantage, which is that physical form can be misleading. For example, a Chihuahua and a Great Dane are two different breeds of dogs, but just looking at them, if you knew nothing about dogs, you may say there's no way that these could be the same species because they look so different. But really, they are the same species. They're both domestic dogs. On the other hand, if you take the Texas coral snake, which you see on the left, and the Mexican milk snake, which you see on the right, they look incredibly similar. But the Texas coral snake is venomous. Its venom contains a powerful neurotoxin, but the Mexican milk snake is completely harmless. So despite their similarity, they obviously don't belong in the same species group. So that's one big disadvantage of trying to use physical form to group organisms by species. Okay, so what about the other species concepts? The second way of defining species is based on the role that the organism plays in the environment, and this is called the ecological species concept. According to the ecological species concept, organisms that share the same niche belong to the same species. A niche is a specific set of environmental conditions that support an organism's livelihood. For example, Charles Darwin, who was the first biologist to ever propose how natural selection works, he studied many different varieties of finches living on the Galapagos Islands, and he found that they had specific habitats and roles that aligned with those habitats. So some finches lived in trees, others lived on the ground. Of the ones that lived in trees, some had a parrot-like beak that they used to eat fruit, others had grasping or probing beaks that they used to eat insects. Of the ones that lived on the ground, some ate cactus, others ate seeds. So they're all finches, but they have different quote-unquote slots that they fit into within their environment, different niches. Where they live, what they eat, how they interact with other species, all of this contributes to their niche. So the ecological species concept says that it is the niche that makes the species. The third and final way of defining a species in biology is also the most common way, and this is called the biological species concept. This is the one that's based on reproductive isolation. So according to this definition, organisms that are able to mate with each other and produce viable, fertile offspring belong to the same species. So in other words, if two organisms can breed together and their offspring are both able to survive, which is what viable means, and those offspring are able to themselves reproduce, then that means that those two organisms belong to the same species. These characteristics of the offspring being both viable and fertile are important because without either one of these things, it means that it's the end of the line for that lineage. So, for example, horses and donkeys are able to mate with each other and produce offspring that are viable. Those offspring are known as mules. But mules are sterile. They cannot reproduce to make more mules. Every mule is the end of the line for its own kind. So the lack of fertility in mules means that horses and donkeys are not considered the same species. They're two distinct species. The advantage of this definition is that it's very useful for understanding evolutionary relationships, which you'll see in just a moment. The disadvantage includes that it's not useful for asexual organisms because they don't mate at all. So this definition doesn't apply to them. And asexual organisms is not a small group because asexually reproduced organisms includes all prokaryotic organisms, most protists, 30% of fungi, 5 to 10% of plants. So this definition doesn't work for any of those. The other disadvantage is that hybridization is common in nature, and it blurs the line between separate species. So hybridization is where two groups of organisms that are thought to be separate end up successfully mating with each other, as in the case of the sika deer and the red deer that we just saw in our checkpoint. And this happens a lot. About 25% of plant species hybridize in nature. 10% of birds have been recorded to mate with other species and produce offspring. So there's definitely some gray area in this biological species definition. But on the whole, it is the most useful definition for understanding how the process of speciation occurs. Speciation is the formation of two species from one original species. So let's walk through how this works using that example of horses and donkeys. Modern horses and donkeys are distant cousins. Evidence shows that they shared a common ancestor about 4 million years ago. However, today we know they're distinct species because they cannot produce fertile offspring together. But how did it get to be this way? So let's say that this little equine figure on the left represents the common ancestor of horses and donkeys. Over many generations, this ancestor would have given rise to many lineages and populations. So in the graphic I've created, I'm showing you just a single individual, but each of these figures is really meant to represent an entire population and many generations within it. So maybe these populations spread out. Maybe they became physically isolated from each other. Some migrated to grasslands, some migrated to the desert. And each of these populations then carries on in isolation from the other populations. Now, some of them will die off and go extinct. Other ones will persist onward, and some of them may splinter into even more distinct isolated populations. And of course, as this occurs, inevitably each population is experiencing mutations over time that get propagated through the generation. And if there's no gene flow happening between the populations, meaning if the populations aren't interbreeding, then those mutations start to build up, and the differences between each population becomes greater and greater. So remember, this diagram is meant to show you millions of years of generational changes happening. So eventually, as the generations carry on and the mutations build up, the remaining populations will become so genetically distinct that they can no longer interbreed. Either their offspring have genetic problems that cause them to die off, or they have genetic problems that make them infertile. And now they're fully reproductively isolated. So this is, in essence, how speciation works. Over many generations, populations without gene flow accumulate mutations that make them genetically distinct and reproductively isolated. Now, do populations need to be physically isolated from each other in order for this to happen? Not necessarily. That's certainly one very common way that this can happen. And when it happens this way, it's known as allopatric speciation. Allopatric speciation is when a population is split through physical separation that ends gene flow between two groups. And there are two forms of this. Dispersive allopatric speciation involves the migration of some members of a population to a new area. For instance, during the last ice age, the fossil record shows that a group of steppe bison migrated across the Bering Land Bridge that, at that time, connected what is now Alaska with Russia. And these became the American bison that we're familiar with today. While meanwhile, the steppe bison back in Asia, whom they had splintered off from, those bison ended up going extinct. The other type of allopatric speciation is vicariant. Vicariant allopatric speciation occurs when a natural event creates a physical barrier that separates a population. For example, the erosion of the Grand Canyon caused a speciation in the regional squirrel population, which is now split into two different species. There's a squirrel community and a species that lives on the north rim, and then there's a separate species that lives on the south rim. However, as I said, physical separation isn't always required for speciation to occur. When speciation occurs without physical separation, this is known as sympatric speciation. Two groups diverge without becoming physically separate. An example of how this can occur is whole genome duplications in plants. There have been many documented cases where plants have, in a single generation, and as a result of a massive error in cell reproduction, made a second copy of all of their DNA, so they went from being diploid to being tetraploid. And then they just carried on with twice as much DNA like it's no big deal. So here's a picture of a normal diploid thale cress on the left-hand side of this photo, next to one that has become tetraploid due to whole genome duplication on the right. These plants are suddenly very genetically different from each other, and they're no longer compatible to breed together. And they could even possibly be physically incompatible for breeding because the whole genome duplication also is associated with a significant increase in size. In fact, the strawberries that you can get at the grocery store are the result of not one, but two whole genome duplications, which is what has made them so much bigger than wild strawberries. Wild strawberries, if you've ever seen them, you can see them on the top. They're tiny little things. If you're ever up on the Mogollon Rim in northern Arizona, keep your eye out for them. You can find them there. But because we know that many plants are capable of self-pollination, even after a whole genome duplication makes a plant instantly incompatible for reproduction with all of its fellow plants, many plants can still self-pollinate and carry out the reproduction, which can lead to an entirely new lineage and species. So this is sympatric speciation. No physical separation required. Time for another checkpoint now. So now that we've learned about the different mechanisms of speciation, which type of speciation would the founder effect be associated with? So we know that speciation is the result of two groups of organisms becoming incompatible for mating with each other due to the accumulation of mutations that make them distinct. But there are different ways that this incompatibility can manifest. Prezygotic barriers is the term used to describe the types of incompatibility where offspring aren't produced at all, meaning that the gametes from the two organisms will never get to the point where they fertilize and become a zygote. That's why these are called prezygotic barriers. They're barriers that come into play to prevent zygote formation. There are also post-zygotic barriers, and those are the things that prevent offspring from being healthy and or fertile. So let's start by looking at some of the different prezygotic barriers. One type of prezygotic barrier is called temporal isolation. This is where two populations have accumulated mutations that cause them to be adapted to different breeding time periods, so they won't interbreed in the wild because they breed at different times. For example, most cacti flowers open during the day and close at night. However, saguaro cacti flowers are the opposite. They open at night and close during the day. So this means that saguaros don't have the opportunity to crossbreed and potentially hybridize with other cactuses. Another type of prezygotic barrier is habitat isolation. This is where two populations have accumulated mutations that lead them to be adapted to different habitats. For example, some populations of walking stick insects prefer to live and feed on California lilacs, which you see on the lower left, while others prefer chamise, which you see in the lower right. If they never visit each other's habitats, then they'll never interbreed. A third type of prezygotic barrier is behavioral isolation. This is where two populations have adapted different mating rituals, and therefore they're not attracted to each other. For instance, many species of birds exhibit elaborate mating dances that would not be recognized by other species. So here we have on the top the blue-footed booby, and on the bottom the bowerbird. Both of them are in the act of trying to secure a mate, but if they were to use these moves on each other, they would definitely not be successful because their mating rituals look so different. There's also mechanical isolation. This is where two populations have accumulated mutations that lead to significant differences in the reproductive organs or structures so that they're not physically compatible for breeding. For example, there are two species of sage in California that can't interbreed. One of them has very long stamens, that's the one on the bottom, and the stamen is what holds up the anther containing the pollen. And because the stamens are so long, these flowers have to be pollinated by very large bees like bumblebees. The other species, the one you see on the top, it has short stamens, so they can only be pollinated by small bees. So although both of these species are sage, they may even occur in the same environment, the structure of their flowers is so different that they're reproductively isolated from each other. The last type of prezygotic barrier is gametic isolation. Gametic isolation is where two populations have accumulated mutations that make their gametes incompatible. Fertilization simply cannot take place. For example, sea urchins reproduce by releasing their gametes into open water and then hoping they bump into each other. However, even if they do bump into each other, the sperm of the giant purple sea urchin cannot fertilize the egg of the red sea urchin and vice versa. They're just not compatible for fertilization. So all of those were prezygotic barriers, barriers that keep two groups reproductively isolated from each other before the zygote has a chance to form. The other type of barriers are postzygotic barriers, and these, as we have touched on, are when offspring can be formed, but they're either not healthy or they're not fertile. Hybrid inviability is where offspring die during embryonic development or otherwise prematurely. For example, goat and sheep gametes can fertilize and a pregnancy can begin, but the embryo will die early in gestation and never makes it to term. And then the other type of postzygotic barrier is hybrid sterility, which is where even if the hybrid offspring survive into adulthood, chromosomal abnormalities prevent them from forming functional gametes. We've already described an example of this in the case of horses and donkeys, where they produce mules as offspring, but the mules are sterile. So, we've come to the end of our lecture on how evolution works. I'm going to leave you with a couple of final checkpoints on these concepts. The American toad, which is seen on the left, mates in late summer, while Fowler's toad, which is seen on the right, mates in early summer. Which type of reproductive isolation is exhibited by these two species of toad? And is this a prezygotic or postzygotic barrier? And lastly, the Mexican long-tongued bat pollinates both agaves and saguaros. Pollen may be carried between the two species, but the gametes will never fertilize. They're not compatible. Which type of reproductive isolation is exhibited here? And once again, is this a prezygotic or postzygotic barrier?